Hello and welcome back to Honors Biology. So up to this point, we've been talking exclusively about types of passive transport: diffusion, facultative diffusion, and osmosis. All of those are from high concentrations to low concentrations. Well, today we're going to be talking about active transport, which is where we move things from low concentrations to high concentrations, because the cell doesn't always need the same concentration inside and outside the cell. Sometimes it needs to get stuff outside the cell where there's a higher concentration because it doesn't belong in the cell. So we're going to talk about active transport. Active transport is different from passive transport in two ways. One, again, like I said, it moves from low energy or from low concentrations to high concentrations. And the second way is that it uses energy. So up to this point, right, diffusion, vacuum diffusion, osmosis doesn't use energy. These next three things we're talking about, they use energy. Protein pumps, ionocytosis, exocytosis. They all use energy. So let's just talk about it. So our first type of active transport are protein pumps. Protein pumps, they transport molecules from low concentrations to high concentrations. So what they do is they remove smaller waste molecules from the cell in kind of a trade-off. So they remove smaller waste products and they're able to get what the cell needs inside the cell using the same energy. So it requires energy in the form of ATP, is what we see down here. And then the example here is the sodium potassium pump. That's the classic protein pump example. So if we look at this picture, this is a prime example of a protein pump in action. Okay, so the white molecules are sodium, green's potassium. So there's a higher concentration of sodium inside the cell than outside the cell. Our cells don't need a lot of sodium, so we need to get it out of there. Whereas you see here, potassium, our cells need a lot of potassium, but there's not a lot out here. So we have to move them against their concentration gradients. So to do that, what the sodium potassium pump does is 3 sodium molecules will hit these little dots here. So they'll come in, they'll bind here. ATP will then come in. So here's our 3 ATP. Here's our energy. We'll release it outside the cell. When it's outside the cell, it'll allow 2 of these potassiums to come in and fit right there. We release our energy and our cell opens back up and into the picture. So that's how a protein pump works, is it moves up against the concentration gradient by adding energy. All right, so that's a protein pump. Our next type is called exocytosis. So sometimes we have really large molecules that just don't fit through a protein pump. So we're able to force things out that are bulky, or just we're able to force a lot of things out. So what it is inside the cell, a membrane-filled vesicle. This vesicle has the same membrane, it's still a phospholipid bilayer that the outside of the cell has. The vesicle will find its way to the cell membrane, where the vesicle will merge with the cell membrane and force stuff outside of the cell. Simple as that. The cell is changing shape because it's getting bigger because we're adding stuff to the cell membrane. So we're increasing the surface area of the cell, and that requires energy. So this is all for hormones or waste products released really from inside the cell. So if the nucleus or ribosomes were to get rid of any waste products, that's how it would do it. So we talked about hormones, dopamine, cytosol. If that stuff to get outside the cell, it excretes it through exocytosis and through vesicles. So cool, that's how we get stuff outside the cell with vesicles, but we can also get them inside the cell through vesicles. And that process is called endocytosis. So all endocytosis is it's taking bulky things into the cell. So all it works is it uses energy, and the cell membrane will fold in kind of around the food particle, right? So it'll come in, whatever the bulky thing is, will kind of butt against the cell. The cell will cave in, form around it, and form a vesicle around it. This is kind of how the cell eats. You can think of this as cell eating. This is actually our idea of how the cell got the mitochondria. So the mitochondria, we actually think was another cell that butted up against a different cell. The cell ate it, went through endocytosis, realized that this mitochondria is a good source of energy, and kept it around. So that's our theory about how we got the mitochondria, through endocytosis. So, and then with the food, again, it forms the food vesicle, and it's able to digest the food that way. And this is actually how we fight infections. So how your white blood cells will fight an infection, a bacterial infection, is your white blood cells will literally eat the bacteria. It will form a vesicle, engulf it through endocytosis. So there you go. So that's it. So that's all there is for active transport. Protein pumps, exocytosis, and endocytosis are 3 ways of active transport. All require energy, and they're all really just for bulky things. So if you have questions, write them down. I'll see you next time in class. Have a good one. Bye.